Good morning and good afternoon. On behalf of IUFRO and the coordinators of Working Parties 7.03.05 and 7.03.16, I would like to welcome you to this webinar in our webinar series on the behavioral and chemical ecology of bark and wood-boring insects. For those of you who are new to IUFRO, the International Union of Forest Research Organizations, this is a global, non-profit, non-governmental science organization. This global organization includes approximately 15,000 members from 120 countries and 600 member organizations. IUFRO is made up of several divisions, and the working parties responsible for this webinar series reside within Division 7, Forest Health. The coordinator of Division 7 is Ecki Brockerhoff. And within the division, there are two research groups, pathology, coordinated by Todd Ramsfield, and entomology, coordinated by Marta Klepwick. These research groups are made up of working parties, and the working parties that host this webinar series, as mentioned, are 7.03.05, the ecology and management of bark and wood-boring insects, and 7.03.16, behavioral and chemical ecology of forest insects. The mission of IUFRO is essentially the same as that of these working parties in this webinar series: to advance research excellence and knowledge sharing and foster science-based solutions to forest-related challenges. One of the many things the current pandemic has made clear to me is that our most meaningful resource is our networks. For those of you who would like to learn more about IUFRO and how to become involved in this powerful research network, I encourage you to consider one or more of the options listed here. Before we get started, a little housekeeping. Josephine Kefelek, a senior PhD student at the Forestry and Agricultural Biotechnology Institute at the University of Pretoria, will moderate this question period. For those of you who would like to ask a question, you can do it one of two ways. You can either use the raise hand feature in the chat box, in which case at the appropriate time, Josephine will unmute your microphone and allow you to ask your question directly of the speaker. Alternatively, you can type the question into the chat box and post it for everyone to see, and at the appropriate time, Josephine will ask your question for you. To make Josephine's job a little easier, I ask that you only post questions to everyone in the chat box, and if you have personal communication you'd like to do, please do that privately. I also remind you that the webinar is being recorded and will be posted after on the Working Party YouTube channel listed here. Before we get started, special thanks need to go out to a lot of people, most notably to Quentin Wignard, who is a senior PhD student also at the Forestry and Agricultural Biotechnology Institute at the University of Pretoria, who manages the IT side of the webinar series, and as mentioned previously, Josephine Kefelek, who will moderate the webinar series, and to the Forestry and Agricultural Biotechnology Institute at the University of Pretoria, or FABI, as many of you likely know it, for providing the platform from which we provide the webinar series. It gives me great pleasure to introduce today's symposium coordinator, Dr. Sigrid Netherer. Sigrid is an Elise Richter Fellow and Senior Researcher at the University of Natural Resources and Life Sciences, Boku in Austria, and with her research team, she investigates the role of drought stress and other environmental and biotic factors on bark beetle infestations to produce a novel universal framework for monitoring and predicting outbreaks. Dr. Netherer is also an IUFRO office holder, serving as Deputy Coordinator of the dynamic and exciting Working Party 7.03.16, Behavioral and Chemical Ecology of Forest Insects. The webinar she has coordinated today involves a series of talks on the chemical ecology of Ips typographus Norway spruce interactions. Thank you.
Hello everybody, my name is Sigrid Netherer. I'm from the Institute of Forest Entomology, Forest Pathology and Forest Protection of Boku, Vienna. Today I will present research about Norway spruce and its interactions with the Eurasian spruce bark beetle Ips typographus. But first of all, I would like to thank Jeremy and Andres and the colleagues of the Bark and Wood-Boring Insect Working Party to offer me this opportunity to present my research today. Let me introduce my co-speakers of today: Anna Jirosová from the Czech University of Life Sciences in Prague. She's a group leader within the quite comprehensive Extemit K project and working about chemical indicators of tree stress, susceptibility of Norway spruce to the Eurasian spruce bark beetle, and about the field activity of new physiologically active semiochemicals. My second speaker today, Raimund Nagel, was doing his PhD and working as a postdoc in Vienna at the Max Planck Institute, Department of Biochemistry of Jonathan Gershenson. Then he went to Iowa State University for some years, and today he is a group leader at the chair for plant physiology at the University of Leipzig. Norway spruce is the most important tree species of Europe, and here in this figure you see its native range extending from Scandinavia over Siberia, Central Europe to Southeastern Europe. Typographus, on the other hand, is the economically most important scolytine species in Europe, and it's also the best studied, or one of the best studied scolytine species worldwide, due to its mass outbreaks, which occur not only in plantations of Norway spruce, but only in the native range of this tree species, so to say in the mountainous and in the boreal habitat of Norway spruce. This figure quite well illustrates the situation we are facing for 30 years by now, starting in the 1990s with large wind-throw events. Since then, Typographus has been more or less constantly in epidemic phase, and this is due also to specific stand characteristics such as high proportion of Norway spruce, high stand age, wind-throw events, as I already mentioned, increasing temperature conditions, and also quite probably due to the increasing frequency of drought periods. When we look at the interactions between the spruce bark beetle and Norway spruce, it is important to understand that both species are part of a very complex multitrophic system that involves a multitude of other species, such as other bark and wood-boring insects, for instance, cerambycids, hymenopteran species, other bark beetle species that are colonizing the same host tree and that are competing for similar niches in that host tree as Typographus. There are a lot of different predator species, parasitoid species, for instance, claret beetles, birds, predator beetles such as Nemosoma elongatum, braconid wasps, flies, and so on. And there's a large range of pathogens, bacteria, microsporidia, pathogenic fungi, mites, viruses, nematodes that play a role in the system. And of course, there are also associated species associated with the spruce bark beetle, symbiotic species. And a very important group of species are the ophiostomatoid fungi that are associated with Typographus. The forest environment, as you see here on the left-hand side, is often quite heterogeneous, mixed with other tree species, non-host tree species for the spruce bark beetle, such as deciduous trees. And it's known that Typographus very well recognizes these non-host tree species and avoids them, but at the same time shows very high potential to successfully colonize its host tree by mass aggregation in response to pheromones. The overwhelming of tree defenses is much easier, as we know, at weakened trees than show impaired defenses. The mechanisms of host selection are not yet fully clear, and it cannot completely exclude it that host choice of beetles might simply due to random landing. However, what we have seen in the recent years is the strong influence of environmental stress on host tree susceptibility to attack, which became quite obvious with the mass attacks observed after heat and drought periods. Conifers, such as Norway spruce, respond to environmental stress such as drought by the closing of stomata, which leads to a reduced photosynthetic activity and also to oxidative and photooxidative stress within the tree. It also induces an increased production of abscisic acid and plant stress hormones, which can be seen in the upregulation of gene expression, for instance. The availability and mobilization of carbohydrate resources for primary and secondary metabolism are kept upright for a very long time and can be seen even in very stressed trees, which still invest metabolites into defense and repair mechanisms. What we also know is that the biosynthetic pathways of specialized metabolites, important for tree defense, are influenced by drought differentially. The effects of environmental stress concern tree attractiveness for the spruce bark beetle, tree defense capability, and host acceptance by the bark beetles itself, and also their colonization success. Looking at the spruce bark beetles, we see that they are very well equipped to orientate in a heterogeneous forest landscape. They can refer to several senses, such as visual senses, so they recognize tree silhouettes in the forest, and they have particularly sensitive olfactory sensilla located on their antennae. In the forest environment, beetles are confronted with negative and positive signals from non-host trees and host trees. It's known from the citrus trees that they have volatile green leaf alcohols and bark alcohols that inhibit bark beetles. So they repel bark beetles, that they don't land on the trees. Beetles show strong neuronal responses to alpha-pinene, which is the main monoterpene hydrocarbon of conifers. And in the field, inhibitory effects have been observed from the oxygenated monoterpenes 1,1-cineol or transphoduionol, the contents of which decrease in trees of higher age. Aggregation pheromone components are the most important components aggregating beetles, such as 2-methyl-3-butanol and also cis-verbenol, which is derived from -alpha-pinene. And it has been shown that there are synergistic aggregation effects of the combination of alpha-pinene with the pheromone components. The associates of osteophytoid fungi have been increasingly recognized in their importance in the recent years. The classical paradigm says that they defeat and exhaust tree defense, that this way promotes tree death, and recently it has also been found that they are able to produce aggregation pheromones, so components at least, such as 2-methyl-3-butanol, and that the fungi can also metabolize terpenes and phenolics for their own benefit. Important tree defense traits that can also be easily measured in the field are constitutive and induced resin flow. This is used by the tree to repel attacking beetles, as you see here in the picture. It can be measured by just collecting the resin over a certain time period in a short glass vial. Hypersensitive food reaction in response to the inoculation of osteomatoid fungi has been often used to assess tree defense capability by measuring the length or the size of the wound reaction zone. You can tell about increased or reduced resistance of the trees. So the shorter, the smaller the wound reaction zones, the higher resistance of the tree. Within the zones, trees accumulate terpenes. And it has also been shown that by mass inoculation, inducible defenses of the trees can be primed. Here I would like to suggest a conceptual scheme illustrating the relationship between drought stress status of Norway spruce and its attractiveness and defense capability against the spruce bark beetle. I want to point out that low, moderate or high drought stress cannot be exactly measured, but is more a qualitative parameter we can observe in the field. Not very much depending on drought stress status, Norway spruce is very attractive to the spruce bark beetles, but what has been on the one hand suggested by several theories, such as the growth, differentiation, balance hypothesis, is the increase of defense capability with the beginning of drought stress, and then the quite quick decrease with increasing drought stress. So Norway spruce is much susceptible, or most susceptible to attack when defense capability is lower than its attractiveness, but when population density of the spruce bark beetle is very low, so the attack pressure is very low, beetles have to focus mostly on the niches of very highly stressed trees. When population densities are really high, we see mass attack of virtually all trees in the forest. The following I present results of my research backing up the conceptual scheme. You see here study plots of the Roselia roof project, which in the first study comprised 3 different treatments: roofed trees, partially roofed trees, and controlled trees, where we measured different hydrological, tree physiological, and tree attack parameters. The second study is still going on, and here we only compared roofed trees and controlled trees. My master student Albert Bachlehner measured resin flow 6 months after roof establishment, and what you see is the clearly increased resin flow rates of the only minor stressed roofed trees. When we look at the results of the first study, 1 year after roof establishment, there's a distinct difference in resin flow rates between roofed and controlled trees or partially roofed trees. But what can still be seen is the induced resin flow at the more stressed trees, indicated by a more negative pre-dawn twig water potential. We used so-called attack boxes mounted at 5 or 6 meter height at the sample trees to study the attack performance of the spruce bark beetles. So we did these experiments repeatedly over the study season, and we always inserted a specific number of beetles from the institute hearing into the boxes, and after 24 hours we looked at what the beetles had done, if they had attacked the tree or not. This might have been successful when we just found a nice boring hole with the beetle inside and some boring dust, or it might have been defended by the tree, clearly indicated by resin flow. The experiments very clearly showed that, as I suggested, tree attractiveness did not differ between the more or less stressed trees, but more or less the same number of beetles were trying to attack the tree. What differed very clearly, especially in spring, was the proportion of successfully and defended attacks of the beetles, with much more successful attacks at the stressed, fully covered trees. We did an inoculation experiment in summer 2020 by use of the fungal species Endoconidia polonica and Cosmania penicillata, and also by use of agar controls. What you see here by the figure made by my master student Giuseppe Morgante is that the more virulent strain of Cosmania penicillata caused much longer wound reaction zones in the roofed trees than in the controlled trees. The roofed trees by that time had been stressed for more than 1 year. We measured total monoterpene contents in the trees, and here are some preliminary results for a couple of trees, which clearly show accumulated monoterpene contents in response to inoculation of both fungal species. To which extent inoculation of ophiostomatoid fungi influences the induction of terpene and phenolic defenses in Norway spruce, and how this relates to bark beetle attack history, will now be explained by Raimund Nagel. But before that, I'm happy to give over to Anna Jirosová, who will tell more about Norway spruce vulnerability for bark beetle attack under abiotic stress conditions as observed within the Extended K project. I thank you for your attention, and later on I will be happy to answer your questions.
Good afternoon, everyone. My name is Anna Jirosová, and today I will talk about 2 research topics that we are studying in my group created within the international project Extended K. First of all, I would like to thank Sigrid for the invitation to present our work and also for her introduction. I really want to thank the organizers of these seminars for giving me the opportunity to meet you all, at least in this way, when it is not possible in person in some international meeting. Norway spruce is a fast-growing tree with good use in construction and industry, which is why our ancestors were establishing spruce plantations throughout Central Europe in recent centuries. In the last decades, the world has been experiencing climate change with the effects of drought and windstorms. The Czech Republic is not excluded. There were several drought periods in 2015 and 2018. In the forests were the most affected Norway spruces because of shallow roots predispose them to be extraordinarily sensitive to drought and wind. Norway spruce's monocultures have been weakened, allowing rapid population gradation of the aggressive Ips typographus. In the Czech Republic, the total volume of trees killed mainly by the Ips typographus exponentially increased from 1.5 million cubic meters annually in 2005 to 2015 to 23 million cubic meters of salvage logging in 2019. The situation affected the majority of regions, and it has not only economical but also a social impact. Extemit K is a project which was founded as a reaction to bark beetle situation in the Czech Republic, and its goal is gather together scientists from multiple disciplines to research not only bark beetle itself, but as well bark beetle Norway spruce interactions. Project is supervised by Professor Fredrik Schlitter from SLU Alnarp in Sweden. Some of my colleagues study beetles on level of genes. They lately published Ips typographus genome, and the other group possesses a broad view on these problematics on level of landscape. Additionally, my group focuses in detail on tree physiology and biochemistry under stress conditions and its relation to bark beetle susceptibility. But we also seek for new biological active compounds in the system Norway spruce, Ips typographus, associated ophiostomatoid fungi. We aim to disentangle chemical ecology principles and find candidates for possible bark beetle pest management. Today I will present the results of 2 experiments we have done in the last 2 seasons. The first one focused on fresh forest sedge susceptibility to bark beetle attack, and second will describe possible biological activity of ophiostomatoid fungi in the long-range attraction of Ips typographus. There are several ways to the sudden formation of the forest sedge. Naturally, this is caused by the windstorms, but it can be created artificially by man if clear-cutting management is applied or after salvage logging. At the resulting forest edges, microclimatic traits as temperature and solar radiation varies, and it results in changes in water regime. The edge of the forests is often the starting point of the spread of the bark beetles. The infestation occurs on the first or second year after the formation of the forest edge. Based on these observations, we proposed an experiment with the main goal to study the development of defense ability and affinity of trees for bark beetle infestation, in connection with tree physiology and microclimatic changes in stands. We set up our experiment in mature Norway spruce forest in 400 meters above the sea level. In April 21st, we cut 4 sun-exposed clearings with approximately 50 meters long forest edges, and we chose control trees in inner forest. There were 3 trees on each plot, 24 trees together. The north and south bark temperature and subflow were recorded on each tree using sensors. Additionally, we collected resin flow and sampled volatile organic compounds by SPME fibers. The soil water potential and meteorological data were recorded on each plot. On the trees monitored in this way, we performed a bioassay with live beetles according to Netherer 2015. From May to August, we performed 7 repetitions. As a first comparison, I will discuss the differences on forest edge versus inner forest in bark surface temperatures measured by apogee sensors. First, I will explain what the numbers above the graph mean. It is a p-value for comparing the differences at the forest edge versus inner forest and shows its significance in a specific repetition. The number below the graph shows the Spearman correlation between the two iterations, indicating whether the property value for each tree are in the same order. Simply put, if the tree that had the highest value on May 7th compared to other trees also had the highest value on May 19th. The bark temperature on the south side of the trees was significantly higher at the forest edge throughout the season. The trees on the north side were also warmer at the edge, but it depended more on general meteorological conditions. Interestingly, the differences in average temperature during the experiment in the south and north side of the trees were only 1 to 2 degrees Celsius at the edge and 0.1 to 0.5 degrees Celsius in the inner forest. When comparing the sum of subflow on the experimental day in the edge trees and in the inner forest, we did not observe any significant preferences. The volume of resin collected in 24 hours was higher at the edge, but the difference was significant only once a season. On the other hand, both traits show high Spearman correlation coefficients, which means that there were trees with constantly higher or lower subflow and resin flow. The soil water potential was recorded by sensors buried 20 centimeters below the surface. The records we present here are the sum of the values in the experimental day per plot. In fact, the soil water potential was high for the most of the season and the trees were well watered. It fell down only in August, more at the edges and close to statistical significance. But even then, it only approached the estimated value for a moderated water stress level. To evaluate the emission of volatile organic compounds as potential olfactometric stimuli for bark beetles, we collected VOCs using SPME fibers. We also took air samples around the experimental plots. To compare the quantity of emitted compounds, we selected the most abundant peaks from the GC chromatograms, which were mainly monoterpenes, as you can see in the table, and we compared the peak areas. The trees at edge emitted more monoterpenes than in inner forest, and the statistic significance was achieved several times a season. In contrary, Spearman's coefficients were very low, which means that this feature were not stable for trees in between repetitions. Correspondingly, when comparing aligned full chromatograms with using a metabolomic approach, we did not observe on PCA plots any separation of edge samples from inner forest samples. Therefore, the profile of compounds did not differ and any so-called markers did not pop up. Originally, volatile substances in the vicinity of the monitored trees were measured either in clearings or in the shade of the forest as background information to tree emissions. In the end, however, the assumption was confirmed that the freshly cut clearings have a high content of monoterpenes in the air, which exceed emission of trees and which contribute or even generate an olfaction niche of the freshly formed forest edge. On this comprehensive matrix are figured Spearman's rank correlations of the courses of previously discussed traits. We can conclude that the resin flow and volatile emissions of trees are relatively strongly correlated with the bark temperature of the trees and also with each other. Subflow correlates with bark temperature, and soil water potential showed no correlation. To determine the actual beetle affinity to trees, or as seen from the other side, trees' susceptibility to infestation by beetles, we employed a bioassay with live beetles inspired by Sigrid Netherer. This is a limited choice experiment in which 20 beetles are placed in a starting bottle on the side of a wooden box with a transparent front wall. Beetles can choose whether to emerge from a bottle, roam or feed on a tree, exit to bottle at the bottom of the box, or, unfortunately, sometimes sneak out and disappear. We evaluated 3 indicators: how many beetles stood in the starting bottle, how many beetles made it to the exit bottle, and how many beetles manifested some affinity to the tree. The number of beetles in starting bottle were lower on the forest edges, and reciprocally there were more exited beetles. In the August repetition, the 2 indicators did not differ. This well describes that the overall moving activity of beetles was higher at the edge until July and leveled out later in the season. The beetle's affinity for the tree is worthy of closer look. Until July 30th, after a 24-hour experiment, we observed only beetles that were hiding under the bark peels. Hypothetically, they would start to feed later, but in time of experiment any boring was not recorded. This behavior was in the beginning of season significantly higher on the edge. However, the real boring activity was noted in the August, when we registered non-significantly higher number of pitch tubes on the edges. This time, the second matrix displays the Spearman's rank correlation of beetles' activity and affinity for tree with monitored traits. The number of beetles in the start bottle negatively correlates with resin flow. Beetles in exit boxes positively correlates with south bark temperature and resin flow. And when we focus on successful boring activity, it positively correlates with bark temperature, resin flow and volatile emissions, and negatively with soil water potential, and it's mainly in the August repetition. We study in this experiment the seasonal development of the fresh forest edge susceptibility to bark beetle attack in correlation to stress events. And the overall conclusion of this experiment is that the trees on the 3-month-old forest edge displayed mild changes in certain defense and physiological traits, which were influenced by season and meteorological conditions. But these trees were not accepted by beetles till the August. And variability in between individual trees in all monitored traits played a substantial role in significance. Here, I would like to present a study that may shed light on the question of whether the ophiostomatoid fungi associated with typographus play a role in long-distance attraction, namely in the aggregation of beetles. It has so far only been speculated. Associated fungi are carried by beetles who introduce them to trees when boring their corridors in the phloem. Our experiment is based on findings of Dinesh Kumar Kandasamy that selected fungal strains when inoculated on agar media significantly attract beetles on 6 cm in a laboratory bioassay. In cooperation with Dinesh, we choose 3 the most active fungi species: Grossmania penicillata, Grossmania europhoides, and Endoconodiophora polonica for testing in a field study. Following the dogma of chemical ecology that the activity of complex stimuli must be demonstrated before testing individual compounds, we prepared fungal mycelia inoculated on agar and placed them in pheromone traps in combination with a low dose of the Ips typographus aggregation pheromone. Mycelia in perforated petri dishes when transferred to the field when they were 4 days old, and the replication was terminated after 3 days when mycelia were 7 days old. We prepared 2 so-called doses. 1 petri dish is called low and 3 petri dishes are called high. The experiment was repeated in 2 seasons during the swarming period of Ips typographus in May. Later, we proceeded to test the compounds which we selected based on Dienish's work, when a mixture of specific compounds emitted by fungi was attracted in laboratory bioassay at high dilution. We prepared equivolume mixture of 3 alcohols, 3-methyl-1-butanol, 2-methyl-1-butanol, and 2-phenyl ethanol, and 3 acetates, 3-methyl-1-butyl acetate, 2-methyl-1-butyl acetate, and 2-phenyl ethyl acetate. Using this mixture, we prepared dispensers with formulated release rate 0.1 mg per day, 1 mg per day, and 10 mg per day. These dispensers were combined in traps with pheromone. After 9 rotations during one experiment, beetles counting and statistical evaluation, we can state that the beetles' catches were upregulated for both EP doses, high GE dose, and high GP dose, compared to the reference value made by agar media combined with pheromone. Catches of low EP dose and high GE dose even showed statistical significance in both seasons. Higher doses were more attractive in GE and GP, while EP was more attractive at low dose. The differences in attraction were related to the profile of emitted volatiles analyzed by headspace from our petri dish settings. We focus on selected alcohols and acetates. According to Dienish's finding, we found the growth of volatile substances in all fungi from the 4th to the 7th day. EP produced the highest content of acetates. GE had the highest content of alcohols, and the total GP emission was the lowest of the 3. When compared with trapping experiment, it led us to the conclusion that a lower dose of these acetates and higher dose of alcohols are important for field attraction. Results supporting this theory were observed when we tested a field equivolume mixture of these compounds. We statistically evaluated that the lowest dose of 0.1 mg per day of this mixture, in combination with the pheromone, has significantly higher catches than the reference pheromone alone. The number of caught beetles decreased with increasing dose. After performing of all these experiments, we can conclude that ophiostomatoid fungi supported the attractiveness of the aggregation pheromone of Ips typographus, and the compounds that play a role are the short alcohols and acetates. Finally, I would like to thank for financial support of our project Extemit K to Ministry of Education, Youth and Sport of the Czech and to European Union, as well as to our Faculty of Forestry and Wood Sciences and also to University Forest Establishment. They helped, for example, with cutting of clearings. There are plenty of people who deserve my acknowledgement. In the Forest Edge experiment was strongly involved my PhD student Barbara Stříberská, who ran majority of it. Then I would like to thanks to Jaroslav Čepr, Miroslav Hradecký, Ivana Tomášková, Rastislav Jakuš, and as well as to Sigrid Netherer. Fungi study was based on cooperation with Dinesh Kumar Kandasamy. But I also want to thanks to Roman Modlinger, to my other PhD student Rajajaran Ramakrishnan, and to my former student Kateřina Berankova. And I cannot forget all the PhD students and technicians from our group who helped in the field and with beetle sorting. And without them we would not be able to run such demanding experiments.
So, welcome to my seminar with the title Does Bark Beetle Attack History Change the Induction of Terpene and Phenolic Defenses in Mature Norway Spruce? This work I carried actually out as a PhD student at the Max Planck Institute for Chemical Ecology with the help of the Zwillenberg-Tietz Foundation, and currently I'm at the University of Leipzig. So the data for this project we started gathering in 2012, and this picture is of the Bohemian forest right at the border between the Czech Republic, Austria and Germany. And this picture was taken in 2012, but the bark beetle attack was in 2008. But as you see, there's still some live tree in the area. So the data for this project we started to collect in 2012, and we used actually trees which survived the bark beetle attack in 2008, and this is a picture of the Bohemian forest right at the border between the Czech Republic, Austria and Germany, and there will be a map later to show you exactly where we collected those trees. But you see that there's always some surviving trees, and the question is actually, why are those trees surviving, or how are they surviving? So for the Bohemian forest we actually sampled those 2 trees and a few others, and the collection was, as already said, right at the border between Austria, Germany and the Czech Republic. So the question actually is, what trees would you use as a control for survivors of a bark beetle attack? Ideally, you would have probed every tree in this forest and then reprobed those which survived. However, as this is kind of unfeasible, we chose an alternative path, and we actually probed a different stand in addition to those which survived a bark beetle attack, and in this stand, which is at a different geographical location, I will show you in the next slide, those trees were already under current bark beetle attack, so the stand was under bark beetle attack. We selected trees which didn't show any sign of a current bark beetle attack, but they might have been attacked by pioneering beetles earlier. And as a control for those, we chose trees of a stand very close, which was never recorded to be attacked yet. So, and for probing those trees, we actually took a little piece of bark at the 0-day mark, and on the other side of the tree, so 180 degrees from this sampling area, we used a cork borer to open up the bark, pull a little plug and put a little bit of A. polonica in, and then after 14 days waiting, we kind of did the same as we did here. We excised a little strip of bark and used it for all further experiments. And for the naming throughout the talk, so PA means generally previous attack, CA means current attack, and NA means no attack. So the second tree stand we probed was in the Kalkalpen, or also in English Limestone Alps, which are located roughly around here. And here, just for reference, where we probed the surviving bark beetle tree, or bark beetle attack surviving trees in the Bohemian forest. And this is how those trees in the Limestone Alps were looking like. And we only probed those ones which still were green. So spruce, as you most likely know, or trees in general, are well defended or can be well defended. And especially for spruce, there's 2 lines of defense. One would be terpenes, which form those resins which are sticky and prevent the bark beetles from entering or can kill them. On the other hand, you have phenolics, and you have one example here. And the questions then become, are those trees surviving because they're better defended to begin with? So do they have higher metabolite levels, or are they able to increase metabolites rather quickly and rather high? Then what is the transcriptional response? How about enzyme activity and intermediate metabolites, especially where we can quantify them? And what about the hormonal response? And I will try to answer those questions first for terpenes, then for phenolics, and in the end I will go into more detail about the hormonal response. So I want to start out with the terpene content, and terpenes are usually classified according to how many carbon atoms are in them. And one major class of terpenes in spruce are the monoterpenes, and up on bark beetle, up on infection with E. polonica, which simulates the bark beetle attack here, trees with a previous attack actually increase their terpene content, as well as trees which are currently under attack and trees which were never attacked. There are slight differences in the amount of terpenes found prior to our inoculation. However, we are unsure how the genetic relationship between those trees is. So we do not know if this is genetics or if this would be a clear pattern. If we then look at the sesquiterpenes, we actually see almost the same trend. Here it becomes a bit more apparent that some of the sesquiterpenes are more abundant in the one area or in the one sampled population than in the other, but overall most of the terpenes are found in all of those trees. And the same becomes true for the diterpenes, and here again we see an increase with inoculation, and we see an increase with inoculation in all locations. So for this levopimaric and palostric acid we cannot resolve on the GC, that's why they're quantified together. So to summarize this, terpenes prior to inoculation differ between the stands, and we are unsure if it's genetics or the history. But all terpenes increase after inoculation, and the pattern is different mostly for sesquiterpenes between those different stands or those different populations. If we take a closer look at how terpenes are in generally produced, so the precursors can be synthesized through two different pathways. One would be the mevalonate, the other the MEP pathway. And then IDS condense those C5 units to longer units, which are the precursors for monoterpenes, sesquiterpenes, and diterpenes. And in plants in general, this is either found parts of it, especially all for the sesquiterpenes, in the cytosol, where monoterpenes and diterpenes are usually present in the chloroplast. So for EDS, we actually looked at transcriptional response, enzyme activity, and metabolite level, whereas for TPS we only looked at the transcriptional response and the end products, which I showed you in the last slide. So if we look at the transcriptional response, or also called the expression profile, and if we do this first for isoprenyl diphosphate synthesis, we actually see there is not a huge increase in transcription. And this is mainly due to those IES actually being very early expressed upon fungal inoculation or methyl jasmonate stimulation. So they already had their peak expression and their expression returned to normal. If you, on the other hand, look at diterpene synthesis, you actually still see an increased expression level. The same holds true for the monoterpene synthesis, and I want to point out especially the delta-3 carine synthase, which increases to about 1,000-fold in trees which are under current attack. But also previous attack or non-attack trees are easily 600 to 900-fold increased. Sesquiterpenes are a bit different, so they already decrease at this time point or show almost no or very ambiguous expression profile. If we come back to the isoprenyl diphosphate synthesis, you can also not only look at the expression levels, but you can also look at how much of the protein or how much of the enzyme is still present in the plant. And you do this by extracting the whole protein and feeding substrates in, so IDP and DMADP, and see what is produced in those enzyme assays. And if you compare inoculated versus not inoculated trees, you see a huge increase in protein activity or enzyme activity, and you see the appearance of another metabolite, which I will go into more detail in the next slide. You can also not only see how much of the protein is there, but you can actually extract plant material and see how much of those precursors, so of those intermediates in terpene biosynthesis, are present in the plant. And you also here see an increase, so you also find more substrates for terpene synthesis. And again, you find another metabolite which is called CDP, and in a chromatogram, you actually see this peak for GGDP, which is the precursor for all diterpenes, and then you see this extra peak, and you find the same in protein assays. And then the question becomes, is it GGDP in a different isomeric form? For instance, it could be all cis, so the double bond would be in a different configuration. However, if you inject this standard with a sample, you see it comes at a different time point. But if you use this normal CDP, so if you use a partially cyclized or a decalin ring cyclized diterpene, which still has a pyrophosphate, you see that it coincides with the peak we find in extracts or in IDS enzyme assays. And so IDS activity is still higher after 14 days, as well as prenol diphosphate levels are elevated, and CDP is only present after inoculation. And to give you a bit more background why we actually see CDP, so diterpene biosynthesis is actually a two-step process. You first cyclize in via class 2 activity GGDP to CPP, so you have this decalin ring, you have a side chain with pyrophosphate, and then class 1 activity produces what we would consider diterpenes, in this case abiotic. And terpene synthesis in conifers are bifunctional enzymes with two active sites. So one reaction, so the first reaction is taking place here, the second one here, which means that metabolites have to travel between both sites. So metabolites are set free before they can be used by the other active site, which means there has to be a pool in the plant, and we are actually detecting this pool. So the other major defense compound are phenolics, and phenolics are in plants mainly produced through para-coumaryl-CoA, which derives from phenylalanine, but can also be from tyrosine. And there is two major pathways in spruce. One involves chalcone synthesis and ends up in flavonols, in flavanols, and the other one is stilbene synthase, which ends up with stilbenes. And those are not the final products, those are just the core structures to illustrate how those compounds differ from each other. So if you then look at the expression profile for those two enzymes I previously mentioned, so for chalcone synthase, you actually see an increase after fungal inoculation. For stilbene synthase, you either see no increase, a slight decrease, or an increase. So this is a not clear pattern in this case. So if you then look at the metabolites, and we will first look at the flavonols, so taxifolin, quercetin, or taxifolin glycoside, you see that there's almost no difference between those, and this is the general core structure for those metabolites. Glycosides means they have sugar side moieties on hydroxyl groups sitting somewhere in those rings. If you look at stilbenes, you see a similar pattern, so astringin, isorapontin, and PCID. You don't see any increase or also decrease in those metabolites. However, if you look at the flavanols, and their general structure is depicted down here, for catechin and gallocatechin, which are monomers, you actually see a difference. Whereas for proanthocyanidin B1 and B2, which are dimers, you do not see an increase. So only monomeric flavanols increase upon an infection, not proanthocyanidin. We also took a look at phytohormones, and in addition to those 4 depicted here, we also looked at salicylic acid, and we were not able to find detectable quantities of salicylic acid prior to inoculation with E. polonica or 14 days post inoculation. So that's why there is no graph about salicylic acid here. But we can either then look at abscisic acid or auxin, also called indole 3-acetic acid, and both of those, which are more growth related than defense related, we do not see an increase. Instead, we see a decrease, which is expected because the plant is under attack and wants to defend itself and doesn't invest material into growing. Instead, we actually find a major increase either in jasmonic acid or into the bioactive compound 7-jasmonic acid isoleucine. So those trees are still, after 14 days, having high jasmonic acid or bioactive jasmonic acid isoleucine levels, which means they're still ready to defend itself and are still investing into defense compounds. So to summarize this part of the talk, we see that fungal inoculation in regards to terpene increases expression of biosynthetic genes. For some genes, we actually pass their peak expression, especially IDS. Instead, here we see increased enzyme activity, we see increased intermediates, and we see more terpene products, and this holds true for monoterpenes, sesquiterpenes, and diterpenes. With fungal inoculation and phenolics, we see that only chalcone synthase expression is increased. Stilbene synthase expression is actually unchanged. And to further emphasize this, or to further have proof for this, only flavanols, which are products of chalcone synthase, are increased. And here I want to again point out that only the monomeric forms are increased, not the dimeric forms. If we then look at the phytohormones, we detect salicylic acid neither prior nor post fungal inoculation. If we look at jasmonic acid and bioactive jasmonic acid isoleucine, we see the content increase. If we, on the other hand, look at IAA or auxin, those contents decrease. But what also becomes very evident is there seems to be no difference between trees that survived a prior or current attack compared to controls that were never attacked. So we still actually don't know what makes those trees, which were sampled after bark beetle attack, after devastating bark beetle attacks, so special. And because this was part of my PhD thesis and I'm kind of switched projects for my postdoc, I at least want to give you a very short introduction what I'm working on now. And I'm still working on phytohormones, but I'm more working on gibberellins. And this is just a short primer. So gibberellins are very essential plant hormones. They are also produced by spruce. And they control growth and elongation. They do so by cell expansion and cell division, and that's their general structure. So this is the easiest bioactive gibberellin. They also promote seed germination or reserve mobilization, and they stimulate flowering. And humans actually use this for malting barley, especially in industrial settings, to increase the size and value of seedless grapes, or to treat cabbage with gibberellins to get it to flowering. So initially, gibberellins were actually not isolated from plants because they have so low abundance, but they were isolated from fungi, and in this case from Gibberella fujikuro or bacanane, which causes bacanane disease, which means rice plants infected with this fungi are actually growing so fast that they can't support their own weight. And bacanane means also foolish seedling disease. So infected plants cannot support their own weight, and harvest losses are usually 5 to 50%, but can reach up to 9%. So during my postdoc, I actually found a biosynthetic operon in bacteria, which is depicted here. and we characterized the operon in multiple bacteria. And if you go through the biosynthesis, you start with N-carine, like in plants or fungi. You oxidize it to N-carinic acid. You then change this ring to form Ga12 aldehyde and Ga12. You introduce a lactone bridge and a hydroxyl group and end up with bioactive gibberellin 4. So gibberellin are. So from knowing the function of the operon, we also took a look at where this operon is actually present, and we characterized it in quite a bunch of those bacteria, and they are present in a quite diverse area of bacteria. So alpha proteobacteria, which belong to the rhizobia, beta rhizobia, and then a huge area of pathogens. And between those, they were most likely transferred through horizontal gene transfer. However, all of those pathogens which belong to the gamma proteobacteria, they are either infecting grass species or cucumbers, and none of these bacteria are infecting trees or conifers. So currently I'm not working on any conifer project, and with this I want to thank the people which were actually involved in all of this, all of the tree projects. So Jonathan Gershenson, Axel Schmidt, Almut Hammerbacher, Michael Reichelt, and Marion Stegel. And then I also want to thank the people at Iowa State, where I did my postdoc on those gibberellin producing bacteria, and my current group at the University of Leipzig, where I actually continue with gibberellin production by bacteria. And then some other collaboration partners and funding sources. And I want to thank you for your attention.
Thank you, Raimund, and for all the speakers for the great talks. So I think we can start with the discussion period. So Raimund, if you're with us, I have a first question for you. So the question is, when you saw the problem in the field, what was the distribution of the surviving trees? And also, why did you decide to evaluate secondary metabolites and also the hormones at 14 days after inoculation?
So, I mean, we only took the trees which were still living. So it's not like we chose who survived and we didn't monitor for survival later, because we inoculated the little plug of fungi and we took it out after we harvested the second time. So we left the trees as is standing, and we decided 14 days because from previous experiments with younger trees that most expression is kind of at its peak or past its peak after 14 days. And we saw in previous experiments, like that's when we saw a good response with hormones, with secondary metabolites. So it was just previous experiments which told us to use roughly 14 days and not wait too long. And we also were asked to not kill those surviving trees. So we tried not to let the fungi grow too long, so we can actually take it back out.
Thank you. So I have another question for you. All the tested trees were still alive. Did you look for signs how intensively they had been or were currently attacked? Maybe these trees were all never attacked.
So we actually checked those trees and they were never successfully attacked. We didn't check, or we don't know if they were ever attacked. This was more of a random selection of those which could have been attacked in this stand. This stand was genetically more conformed than what was in the Bohemian forest. So we randomly selected trees which were interspersed in those dead trees.
Okay, thank you. I think I have another related question. Have you tested mechanical defense such as bark thickness?
No, we have not.
Okay, thank you. The next question is for Anna. Were there also natural attacks at the forest edge or the sample trees within the forest? And did this show a similar pattern with regard to timing and tree preferences? We can't hear you, Anna. Your mic is not on.
For question,
Actually, we didn't notice the natural bark beetle attack on our fresh forest gist. We will go to check this year. Maybe this year it will be infested already. And the second part of question was, I'm sorry.
Yeah, sorry. Did these show a similar pattern with regards to timing and tree preferences?
Actually, it did. The trees were kind of constant. When we saw the first real attack, it was in all the trees together in April, actually.
Okay, thank you. I have another question for Raimund. There did not seem to be much of a pattern in the chemistry between the trees in your different treatment groups. So can we conclude that the survival of trees interspersed with dead trees more related to the aggregation behavior of the bark beetle and therefore more a random process than related to tree physiology?
I would tend to say yes, but I think the sample number is a bit little for this. So we actually really more tried in this to have all the secondary metabolites, have transcription, have phytohormones, basically have on a set of tree everything done. But I don't think we have enough numbers to say this is random. And I mean, for random, it would be ideal to have a stand, survey them for years, keep samples for years, and then in the end, see who was attacked and who was not. But this would be extremely long-term and extremely work intensive.
Thank you. Another question for you. I am most surprised by the decrease in ABA after induction. This is often induced in response to attack and associated with defense gene injection. Can you comment?
I think it's in some species, ABA is more associated with some attacks, but I think there's also a difference between necrotrophic, biotrophic, hemibiotrophic. So I'm not 100% if the fungi will model the beetle exactly, but that's at least what we saw. And I think we saw this before in small trees where we sprayed with methyl jasmonate that ABA also went down, but I would have to actually go back to the data.
Thank you. The next question is for Anna. Do I understand it right that there is not much difference in the tree physiology based on the location of the trees? Would you thus conclude that the lack of trees is related to the... Oh, no, sorry. I get confused there. That's not for you. Under what natural conditions do you expect that bark beetles will respond to fungal volatiles?
Thanks, Nadir. It was a question from Nadir, I guess. Actually, we started this experiment with the hypothesis that bark beetles actually infest the volatiles to their corridors. So the first are the males, and then they are calling for conspecifics. That means for other males and also for females. There is also known the sequence that first is growing Endoconodiophora polonica in the corridors, and the other fungi, mycelia, is developing later. So it can be also connected to the real ecological or chemical ecological function of these fungi. That, for example, Endoconodiophora polonica could have a more higher level of attractivity because it's growing still in the fresh tree in the beginning of the attack.
Thank you. I think I got confused with Marsha's questions. I'm very sorry. Okay, I have a question for Sigrid. How did you make sure that the roots for the spruce under roots did not receive water? I guess they didn't see your results, but did you try to measure that in some way?
So actually, we cannot completely exclude that the roots got some water. And this was also not the aim of the whole study, that we completely depleted the spruce trees of water, because this would have led to a quite quick decrease in vitality, and I guess a quite quick dieback of the trees. What we wanted to induce is a slow stressing of the trees. So, and the fully covered trees in the first experiment, maybe you remember from my presentation, we had 3 different treatments: fully covered and partially covered trees and control trees. And here we saw really nicely that the trees where we had the large roofs around in the second year were quite severely stressed compared to the control trees, and also the only partially covered trees that were only minor stressed due to the weather conditions. And in the second part of the study, we therefore decided only to roof or not to roof the trees. And because the weather conditions in the last years, so in 2019 and 2020, were not so hot and not so dry, we only received a minor stressing of the trees, which actually brought quite interesting results when we looked at the different defense traits. So we saw that at least in the first year of the study, the defense of the minor stressed trees was increased. But we know from the first study run that when the trees get more stressed, then the defense goes down.
Thank you. I have another question for Raimund. Was the age of the trees an issue?
They would have all been well in their prime for being attacked by bark beetles, so that they were all of a similar age group, which is attacked by bark beetles. So we don't think they were, and the tree stand was of a very similar age. So most likely not.
Thank you. Another question for you. What other variables could have possibly affected the tree physiology in order to get no correlation between bark beetle infection and tree physiology?
So we didn't took any other tree physiology into account. So we only, especially Bohemian forest, we only took survivors, and it was a special permit to get into this kind of, it was not the core zone of a natural preserve, but it was close to the core zone. So we had to ask foresters to actually bring us there, collect the samples, and this is kind of involved a hike. So we didn't also wanted to do it multiple times. So we didn't really record all of the tree physiology of those individual trees.
Okay, thank you. Question for Anna. Did you see a sex-specific response of Ips typographus to fungal odors in the field? Your mic is off again.
Yeah. Thank you, Martin. Actually, we were really curious if it will influence the sex ratio. And just now we finished with the sex sorting. I was kind of disappointed because for the fungi which were actually inoculated on agar media, we didn't see statistically specific difference in ratio of females and males in catches. There were some differences in the part of the experiment which I didn't present here today, when we actually inoculated fungi or on agar with phloem. But again, there played a big role when it was collected in the seasonal time, and also, for example, it was different during a day. That they were also a diurnal influence. So now I cannot see that we saw the really strong effect of fungi for a sex ratio.
Thank you. Another question for you. Did you see a response of predators and other associated insects to the fungal volatile?
Yeah, it was also what we wanted to find out. Actually, we didn't find too much predators. We found like really individuals, because actually the bark beetle traps are not suitable, for example, for flies. Or other flying insect. So next time when we will set up similar experiment, we probably should add some sticky traps nearby, and then we will know more.
Okay, thank you. The next question is for Raimund. Do you think how fast trees respond to bark beetle attacks makes a difference in terms of outcome of attacks by beetles?
Yeah, I certainly think so, but it might also be the luck of the beetle to hit something in the bark, like hit a resin channel and be unlucky to be flushed out or die in it, versus some bark beetles which are not actually hitting those and can colonize first, and then so it might also be a lot of chance to this. And I think there was something else for physiology. It might also be, or what we actually expected is to see in those surviving trees, extremely well defended trees which have extremely high amounts of resins, phenolics or something, versus others which basically have very little and go under the radar, so they're invisible for the bark beetle. But as it turns out, yeah, they're all almost the same. And we looked in the data for like outliers, which are extremely high or extremely low, and they were completely normally distributed. So there was there was a very normal and very narrow range compared to other trees we have, which are clonal.
Thank you. Next question for Raimund again. Is anyone currently investigating genetic effects on the production of terpenes?
With genetic effects, I'm not sure if they mean like in whole tree stands, but I'm not aware that people are looking into promoters and which tree, which specific has what. So there's a recent review on which terpene syntheses are in conifers, especially I think pines from 2012 or 21. 2020 or 21, but I don't think there's any kind of which genetics is superior, which is not, and which terpene synthase is in which kind of variety or in which specific tree.
Thank you. Next question is for Anna. Would you say that the attack of trees is related to the bark beetle behavior more than location-related tree physiological responses? Your mic is off again.
There were a very similar question for Raimund a while ago. Actually, in our case, it will be probably both traits. There will be some part that trees are under the stress conditions and they really change the physiology. We observed mild changes, but because, as you noticed, our trees didn't step up the border of severe stress through whole experiment. But sure, there will be also a reason on the side of the beetles. There were a lot of work published that actually there can be just that the fresh forest, which is just the first obstacle during their dispersal flight, and that they just hit to this, like to the first location, or that olfactometric issue, like high, or in the beginning it's too high, so it's have to go lower. The amount of monoterpenes in the air can trigger the attraction of the fresh forest edge. And also the temperature itself is playing a role, because beetles need some optimal temperature for moving and for developing as well. So it can offer the forest edge for them earlier, especially in the spring, that in the inner forest. So yeah, we can discuss here a long time about what is the cue, or which cue is the more important for the fresh forest edge are the initial location for bark beetle attack.
Thank you.
sorry, maybe I'm allowed to add an interesting observation we had some years ago in a master thesis when we looked at the forest edge that was caused by an avalanche. And these trees, some of them were quite severely hurt by the avalanche, so they had big bark wounds. And we saw that they quite significantly increased the resin flow in response to this wounding, and that they were not attacked till the end of the season. Then they started to be attacked. But the trees that were first attacked were actually a little bit more inside the forest. So this wounding in the first hand caused an increase of the defense. And maybe I think, Anna, your results point a little bit to this direction, that this happened here again. That they became vulnerable over the season then to the attacks. The trees at the edge.
I would say so, because as I showed the results from the also monoterpenes in the vicinity of the forest edge, they were really high in the beginning, like directly after the tree cutting, which is kind of obvious. But maybe the smell was such strong that beetles were even repel and not attract in the beginning.
Thank you. So the next question is also for Anna. The buildup of biomass of ophiostomatoid fungi in the tissues of live trees will take considerable time. Can their metabolites have an influence during the early phase of attack by Ips typographus when their mycelial quantity is still at a low level?
Thanks for this question. I know that it takes time. It's about 3 weeks actually. And as I mentioned before, there is some sequence that some fungi is growing faster than others. Sure, it have to take some time when mycelia are grown enough that they will start to emit enough volatiles, which actually can be even attractive for Ips typographus. But as I told before, it also can depend on the sequence, which fungi is the faster grower and which is the slowest. And then it can depend which volatiles is produced the fastest one and which is produced the slowest one.
Thank you very much. I think we're running out of time and Jeremy has a few announcements to make.
So first, I would say thank you very much to Sigrid, to Raimund and Anna for a fantastic webinar and a great end to a successful series. Just before everyone goes, I wanted to say thank you all for joining in all these weeks. This is the last webinar. The next steps for the working party and for the division, there will be an all-division webinar series that will start in September that is designed to address topics that are overarching for pathologists and entomologists, trying to bring the communities together. So please watch for announcements about that. Within the working party 7.03.05, 7.03.07, and this one, we are going to be launching a mentoring network that is designed for early career researchers and graduate students to try to help them develop networks and soft skills. So please also watch for that. If you're interested, Justin Whitehill from NC State University will be coordinating that activity. So that's everything for now. Thank you all and have a happy Easter.
Bye, everyone.
Bye.
Bye-bye.
So I don't know if,
Thanks for all the hard work.